Hello, everyone, and welcome back to part two of the All Protein Dialogues webinar, Understanding Nutrition in Alternative Proteins, Insights from Across Asia and Beyond. My name is Amrutha Girivasan, and I'm part of the SciTech team at GFI India. I'm thrilled to be hosting today's session as we continue to deep dive into one of the most important topics in the all protein space, which is nutrition. This two-part webinar is part of the broader All Protein Dialogue series, a collaborative effort by GFI APAC, GFI India, and GFIC to spotlight key issues and innovators shaping the future of alternative proteins across Asia Pacific, India, China, Australia, and New Zealand. Last week, in part one of this webinar, we explored nutritional formulation, biofortification strategies, and ways to enhance nutritional quality in alternative protein ingredients and products. Today, in part two, we'll hear from two experts, Dr. Han-Yan-He, Associate Professor at Shaanxi Normal University, and Dr. Jia Yee Wu, Scientist at A*STAR, on their work in enhancing nutrition in alternative proteins. I'd like to take a moment to thank our speakers for graciously accepting our invitation to speak at this webinar. Each speaker will present for about 20 minutes, following which we'll open the floor for a 15-minute live Q&A where you can ask your questions directly. Please use the Q&A box to send in your questions at any point during the session. Kindly make sure to mention the name of the speaker that you want to direct your question to. And additionally, do keep an eye out on the Zoom chat where our team will share helpful resources and links. This session is currently being recorded. With that, I'd like to introduce our first speaker, Dr. Jia Yee Wu, biomedical scientist at the Agency for Science, Technology and Research, A*STAR, with joint affiliations at the Singapore Institute of Food and Biotechnology Innovation, SIFB, and the Institute of Molecular and Cell Biology, IMCB. She's passionate about advancing research at the intersection of nutrition and translational medicine to promote health. Her work focuses on understanding protein quality and its influence on metabolic health. She leverages advanced proteomics, particularly mass spectrometry, to uncover the molecular mechanisms through which nutrition can drive positive health outcomes. Dr. Wu's talk is titled, Are All Proteins Equal? Insights from In Vitro Digestion and Human Studies of Alternative Protein Sources. Dr. Wu will explore how we can use proteomics and peptidomics to understand alternative protein nutritional properties and then move to two case studies of all proteins in human trials. Dr. Wu, I'll hand it over to you now.
Thank you. I'm sharing my screen. Can I just quickly check if you have already seen? Okay, cool.
Yes.
Okay, so first of all, thanks for the GFI team for inviting me today. It is really a pleasure to be here in these sections with people across the world. So the focus today is about alternative protein sources, protein quality, and about nutrition. Yeah. So before I start, I would like to introduce myself and the team where we are from. So we are from Singapore Institute of Food and Biotechnology Innovation, SIFB. SIFB is a translational institute with core capabilities in synthetic biology, food science, of course, biotrans, engineering, so also nutrition and digestive health to develop, so our aim is to develop innovation solutions for nutrition and also in the sector of consumer cares. Yeah. So before we jump into all sorts of different proteins, I would just like to spend some time to talk about why are we studying dietary protein? Why is it important? So essentially, can I ask if you can see my cursor?
Yes, we can.
Okay, cool. Yeah. So essentially, we eat to get nutrients from food, right? The ultimate goal is to really get energy for our cells energy use. So ATP is the currency of the energy in cells. All sort of the nutrient source, such as like carbs or like lipids, fats or proteins, they are all capable to become acetyl-CoA, which is a source to be used by cellular respiration to generate ATP. But protein is special in two ways. First of all, if you look at glucose or any fatty acid, in our body, we have a storage for this kind of nutrients, but the only exception is for amino acid. So we don't really have a storage for amino acids. When we eat more protein that we need, it is immediately excreted out in the form of urea, which is why having enough proteins becomes a very important topics in the field, especially in nutrition. Also, besides being the source of the cellular ATP, amino acid also has another role, which is to be a building blocks of our body. Yeah. This is why it is really essential. Yeah, so this is a predictions of how the alternative protein market will continue to grow. So regardless we are ready or not, whether you like it or not, whether you like the protein on the shelf close to a supermarket, it is becoming a fact that the alternative protein market is accelerating. So for us like food scientists, it is very important to understand what is the potential impact for consumer switching from conventional protein sources to alternative protein sources. Yeah. So ensuring nutritional adequacy is a multi-level, multi-layer perspective. So you can imagine that to get these nutrients into my body, first we need to have the protein accessibility, right? It need to be accessible in the region. And then we need to get people to go and buy it and eat it. So then it comes appetites, it comes to the play, whether they have the good appetite or like whether the food is appealing for them to would want to consume is another layer of the control. Then after we eat the food, whether these proteins has enough amino acids bioavailability, whether it is digestible or whether we are able to absorb the proteins nutrients becomes the next problem, right? And then at the end, after we eat the proteins are digested into amino acids circulating in our body. Is our tissues able to uptake this type of nutrients into the cells? Then this is again another perspective into the nutrients. Yeah, so to ensure nutritional, especially protein nutrients adequacy is actually a multidisciplinary approach. So it's not just about nutrition, it's also about the food science, food security, and it is something we need to work together to address the complexities. Yes. Yeah, so in the research settings, how do we evaluate protein qualities? I listed some commonly used method. So one would be to directly assess the digestibility. And then the other way would be we feed it to human, then we check how are these proteins, so we track how are these proteins turned into amino acid and where is the fate of this amino acid in the body. So today mainly I will focus on the digestibility part. And for digestibility, there are two scores that are commonly being mentioned. Perhaps you heard about it. So currently it's becoming more and more popular. I've seen like people who are really keen on building muscles, they really start to look into the PDCAS and the DIA score for the amino acid. Yeah, so what is PDCAS? PDCAS is essentially a very basic way, I should say a very simple way to measure how digestible is a protein. So protein in, protein out. So they measure the protein content before you consume the food, and then also again in the fecal. So you know that the protein in minus out is the one that retain in your body. So that's how they figure out the digestibility of the protein. But this is being criticized for two reasons. One is that it measures nitrogen as the protein score. So sometimes if you have other nitrogens compound in the food, then it might not be accurate. And then second thing is that it measure at the fecal, but actually what happened in the digestive tract is that after ileum, the digestions become the colon fermentation, which doesn't have much of the I should say after colon fermentations, then the bacteria come into play, which is not directly measuring the protein digestibility, but also the effects of the bacteria. Yeah, hence there's another score which is called 𝗲𝗂𝗃. So 𝗲𝗂𝗃 is quite similar to 𝗶𝗃, but there's a bit of difference. So it measures ileal digestibility rather than fecal digestibility. Also, rather than taking the whole protein content as a whole, it actually gives score of each amino acid, and especially those that are essential, the indispensable amino acid profile. So I just talk about digestibility and how can we turn this digestibility into the protein score. Yeah, so basically there has been already a group of experts. They have already crafted a benchmark, which is called the benchmark is a mark that a score that measure our body daily needs of the protein. Yeah. So if your protein sources, if you look at this ring, if your protein sources meet the benchmark, and then your protein score would be 1. 100% would be 1. But if any of your amino acid doesn't meet the requirement, let's say it meets 60% of the requirement, then your protein score would be 0.6. So as you can see, this kind of scoring system, if you want to hit a higher protein quality score, you need to increase the limiting amino acid. You need to bring up the limiting amino acid. So how can you do that? Is either by increasing digestibility or increasing the limiting amino acids. Yeah. So here there's an opportunity here because people would want to boost, especially from the commercial side, they would like to boost their protein score of their raw protein sources. So the opportunity is to address this gap, the gap here. So how can we bring up the digestibility, or how can we give to supplement the limiting amino acid? Yeah, so to do that, there's only one way, which is to profile and understand what is exactly happening in your protein source after digestions. Yeah, so here I would like to introduce our pipelines on how we do that. Yeah, so this is a in vitro digestions peptidomics. So what happened is that usually in digestibility, after you consume the food are being digested by the enzyme actions into smaller pieces. So you can imagine that actually in the digestome, you have a snapshot of the complex profile. So in this complex profile, there's a mixture of readily absorbable amino acid, free amino acid. There might be di or tripeptides, also readily absorbable. There might be longer peptides. There might be peptides that are just resistant to be broken down, or really a protein fragments that cannot be break down. So in this complex profile, what I'm interested to see is that is there any insights we can get from these undigested peptides or proteins, and can we use this information to understand what exactly happened and how to improve? Yeah, so this is just some example of what you can get by profiling these peptides. So we can look at the size distribution. If the size is towards the smaller size, that means the protein are in general very good, very digestible. But where is if your protein is on the larger peptide length, that means the unlikely this protein will be absorbable. So this is one information. And then also with the power of the peptidomics, we can also look into the size of fragments and know exactly which fragments is the one that causing issues. We know exactly at which amino acid, at which proteins domain the indigestibility happens. Also, we can also look at the mapping of the amino acid, and sometimes we will see some bottlenecks that blocks the digestion. So this is an example where a proline at the second amino acid is always blocking the trypsin actions, which makes the peptide not digestible. So yeah, so it might be a bit technical, so bear with me. I don't want to go into so much detail, but essentially I just want to mention that this is being done using in vitro digestions, and then we are using LC and mass spectrometry based method to profile these peptides, and then using an untargeted approach. Yeah, so I will share some result from this approach. Yeah, so basically we use InfoJazz as our in vitro digestions. So InfoJazz is a very powerful tools because it's internationally harmonized. So in a way, the result, the data you have is directly comparable to the data from another labs. So this is a very powerful tools. And it has been actually validated. They have compare the in vitro digestibility to the in vivo, and it seems that they are in good agreement, which makes it a good tool to use. Yeah. So after having this pipeline, the first question we ask is how is plant-based protein digestibility compared to the conventional animal-based protein digestibility? So the first thing we do is to this is a comparison of chicken. So we have chicken breast, and we have something more processed, chicken balls. We have then from plant-based, this is a common formulation for mock meat. So here we have a formulation which is 40% soy protein and 60% wheat gluten. And then here you are seeing the peptide length distribution of the digestome. So you can see that for chicken, mostly the peptides are centering around 10, a bit plus minus 5. Yeah. But when it comes to the plant-based, we see extra long peptides in the digestome, indicating that they are not being properly digested. Yeah, then we were wondering, actually this is a bit, when I get this result, we were like surprised. I mean, we know that protein digestibility is not as good as animal, but we did not expect to see such a difference. Then we were wondering, why is that? Was it because of the processing? So here there are two different processing for this mock meat. One is through the extrusion and the other one is through mechanical elongations. So to understand whether this is a processing issues, we also include a non-processed food metrics. So just profiling the unprocessed powder, it seems that I am again getting a similar profile. Yeah. So what happened is that there is a if you look at the peak, there is one long peptides here at around 32 amino acid length. So with this power of the peptidomics, we can look into this peptide, and then it's confirmed that this peptide, we know that it is from soy, we know that it is from a glycine G5, and we know that this is an amino acid from 305 to 336. So it's this fragment that is giving us problem. Yeah, so this is the beauty of these two, that not only it tells you where the problem is, it directly tells you potential ways to solve that. Yeah. So again, we also look into other higher peptides, higher length of peptides, and then we discover that actually gluten is giving some problem. It causes really long peptides with glutamate repeats. So after discussing with my colleague, food scientist, he then tried another plant-based meat, plant-based mock meat formulation. So here, instead of wheat gluten, we switched to pea protein isolate. And as you can see that there's two different productization from these formulations, and these are both Asians food. Wonder if you recognize that. So from there, we see that the problem seems to be solved because now the peptides are all left shifted, making them more digestible. Yeah. Yeah. So based on this mini study, it seems that regardless, I shouldn't say that, but I mean, protein source, it seems to play an important roles in deciding digestibility. Okay. Processing could play a role, but is not as impactful compared to the protein source. So this tells us that if you want to make a protein, plant-based protein or any other alternative proteins with good digestibility, to start with the right protein source is the most important thing, because the rest of the texturization, it wouldn't change much on the profile. Yeah, so this is what we discover. I mean, we have only tried these these conditions so far. They are much more different type of processing, and of course they might have effects on the digestibility, but I just don't know yet. Yeah, so after getting there, we are also interested to look into other sort of protein sources. So I would just guide you to here, look at alternative protein sources. It seems that mushroom is in general quite good. and 𝗲𝗲 is a type of seaweed, and it seems to have moderate peptides generated, but in all they are quite okay, I would say. Like there's no not unreasonably long peptides being found in the digestome. So they are all quite promising protein sources to be used in incorporating into our daily lives. Yeah. Yeah, so because of the time constraint, I will go super fast later, but so this is the end of the peptidomics. So I just want to say that this is a tool that you can use to profile the digestibility. So next, I just want to quickly jump into some human studies we have done and quickly show the outcome. So as a conclusion, I would just like to summarize that InfoJS combined with the mass spec-based peptidomic analysis is a powerful tool for us to access nutritional quality of dietary proteins and also at the same time to gain insights on how to improve the protein quality. And then from the human study of the seaweed, the nori protein biomass has the potential to be the functional food for lowering triglycerides. And then if you have good optimization, plant-based protein is actually as superior as animal-based proteins in terms of the nutritional properties and also acceptance. And sometimes it can even do better for the metabolic profiles. But I would like to emphasize that this really depends on how you make your food metrics. Yeah. So there is no direct or black and white yes no. So it really depends on the protein itself. Yeah. So I think this is what all food scientists and nutritionists can work together to ensure that the products on the market are all of the better qualities. Yeah, I will end my presentation here. Thank you.
Thank you so much, Dr. Wu, for your wonderful talk on in vitro digestion and human studies of all protein sources. I think understanding how all protein sources and the resulting products behave once they are being consumed is becoming increasingly important, especially as we pivot to more sustainable diets. So if anybody has any questions for Dr. Wu, please drop them in the Q&A box with Dr. Wu's name so that we can take them at the end of the webinar. Next, I'd like to introduce our final speaker for the webinar, Dr. Han Yan-hee, Associate Professor of Food Science at Shaanxi Normal University. He earned his PhD in food science from the University of Massachusetts Amherst. Dr. Han's research focuses on the interactions between food components and gut microbiota and the development and application of plant-based proteins. Dr. Han has published over 30 peer-reviewed papers in top-tier international journals, including Trends in Food Science and Technology and International Journal of Biological Macromolecules, to name a few. Dr. Han is an active member of the Institute of Food Technologists, IFT, the American Chemical Society's Agricultural and Food Chemistry Division, ACS, and the American Society for Nutrition, ASN. Dr. Han's talk, titled The Nutritional Promise of Mycoprotein Components, will highlight his work on investigating the effects of different byproducts from the food industry on the production and properties of mycoprotein. His research compares mycelial protein yield and analyzes key physicochemical and functional characteristics, highlighting how substrate selection influences the quality and the potential applications of mycoprotein in food systems. Dr. Han, I'll hand it over to you.
Hi. Is it clear?
Yes, we can hear you.
Okay, yeah, I'll share my screen. Okay. Okay, is the screen being shared?
Yes, we can see it now.
Okay, yeah. Okay, thank you. Okay, yeah. Hello everyone, I'm Qian Han from Shaanxi Normal University in China. Today I'm very pleased to share our recent research on the nutrition potential of microprotein-related components. Here, with the increase of world's population, the demand for protein is increasing dramatically. However, the consumption of traditional meat linked to some healthy problems, like the long long-term consumption of red meat, particularly processed meat, raise the risks of many diseases like mortality, cardiovascular disease, colorectal cancer, and type 2 diabetes. Also, the consumption of traditional meat raises some concerns about environmental problems and animal welfare. As a result, the protein alternatives are attracting public attention now. So the plant-based proteins and the cell-based meat are the hotspot in this area, but both of them have limitations. Most of plant-based proteins are intractable because of their inferior functionalities. Also, their low protein quality and undesirable flavor significantly limit their applications. For cell-based meat, their production scale and the cost of cell culture medium also significantly limit their commercialization. So beyond the above two options, mushrooms are reaching. Mushrooms is another promising source of proteins, and they have many advantages. Like firstly, mushrooms are rich in high quality of proteins. These proteins contain all 9 kinds of essential amino acids, in contrast to other plant-based proteins, which typically missing 1 or more essential amino acids. Secondly, the flavor of mushroom protein is more acceptable than bean flavor. And thirdly, the fibrous or flaky texture of mushroom protein is similar to the meat fiber. And even more promising is the mycelium. The protein content in mycelium is even higher than those in their protein body. For example, here the 𝗲𝗵𝗴𝗀𝗀𝗲𝗃 urinocysts. In their protein body, the protein content is around 20%. But in the mycelium, the protein content can be double that. So if we can use the mycelium as protein producers, this approach may show good opportunities that be faced with other protein alternatives. So far, the microtechnology food ingredient company, they have made a successful protein product named microprotein, and the protein is from Fusarium vignetum. So we can see here, utilizing the mycelium as protein alternatives may be a feasible approach to promoting the production of protein alternatives. So in our study here, we focus on 2 aspects. First, we try to screening edible fungi to identify suitable and optimal strains for the microprotein production. And secondly, we try to utilize the food industry byproducts such as citrus peels, apple pomace, and soybean meals as fermentation substrates. Here we choose young apples. It is a kind of waste during the thinning period of apple growth. So we can see we try different kinds of fungus. We can see here from the left figure, the young apples can be totally utilized by the fungus with the production of mycelia. We can see their structure or morphology of these mycelias. So by evaluating their biomass yield and crude protein content, also the amino acid profiles, we choose Schizophilum here, this fungi commune as optimal edible fungi for fermenting young apples to produce microproteins here. Following the successful production of microprotein, our research took 2 directions. Firstly, we try to develop meat alternatives use produced microprotein. And secondly, we try to explore the functionalities of this microprotein for their health promoting benefits. Today, I will focus on the second area, particularly the bioactive functionalities from the microprotein-related components here. So as a novel protein, so we try to find some new bioactive peptides from the microprotein. Here, after hydrolysis, thousands types of peptides have been identified. So through the bioinformatical predictions, 3 types of peptides, as shown here, the structure of these 3 peptides. These three kind of peptides show the strong anti-inflammatory effects. So nextly, we try to investigate the anti-inflammatory effect of these 3 peptides. So in the left figure here in the normal cells, these 3 peptides did not show any cell toxicities in the normal cell lines. In the inflammatory models, as we can see in the right figure here, with the increase in concentrations of all 3 peptides, they significantly increase the inflammation. Here we can see. Also, this inflammatory effects show the dose-dependent manner. And next, we also found that the 3 peptides treatment can significantly inhibit the inflammatory marker like the inos, as well as the inflammatory cytokines like interleukin-1 beta and interleukin-6. And additionally, the inflammation can cause abnormal cell apoptosis, as we can see in the upper figures here. The inflammation causes significant amount of cell apoptosis compared to the regular ones. However, the peptide treatment all 3 peptides significantly inhibit the abnormal apoptosis induced by the inflammation, particularly the effects of higher concentration of these peptides here. So this part of our study is about we try to figure out, try to screen some novel peptides from the microprotein, and this part of work being reported already. So another exciting area is the discovery of exosome-like nanoparticles released during the fermentation. So during the submerged fermentation, we prepare the microprotein. With the growth of mycelium is associated with large amount production of nanoparticles. These nanoparticles rich in lipids, proteins, nucleic acids, and other biotic components resemble the exosomes found in plant. And the emerging evidence suggests that these nanoparticles may offer health-promoting benefits. So we try to isolate and purification these nanoparticles from the fermentation progress. And this is general progress how to obtain these nanoparticles. And this progress mainly involved in the centrifugation, ultrasonification, and the purification by sucrose. Here we can see the clear band of these nanoparticles here. And also we characterize these nanoparticles. Here the DLS and NTA are usually 2 ways to detect the nanosize of these particles. So we can see from these 2 methods, the results show the similar particle size distribution. Also, the zeta potential analysis indicated that these nanoparticles show the negative surface charge. And from the SEM and the TEM, we can see these nanoparticles showed spherical and cup-shaped morphologies. We obtained these nanoparticles from the submerged fermentation. We try to figure out whether they have some bioactive functionalities. So here we mainly focus on their interactions with the gut microbiota. So firstly, we try to figure out whether these nanoparticles can be internalized by gut microbiota. We can see from the upper figures, we can see part of the gut microbiota can uptake these nanoparticles. So next, we try to figure out what kind of bacteria can uptake these nanoparticles. So these nanoparticles have been labeled with dye, and the bacteria being screened through fax these bacteria being also labeled with also show dyes because of their internalization with these nanoparticles. Then this bacteria were being further being determined. So our results showed that some bacteria can uptake these nanoparticles, including Bacteroides, Lactobacillus, and bifidobacteria. So here we mainly focus on the well-known health benefits, health promoting gut microbiotas. So another long-term study also confirmed our results. Here we mainly focus on the beneficial bacteria like Lactobacillus and bifidobacteria. As we can see here, the abundance of lactobacillus was significantly increased with the treatment of microprotein-derived nanoparticles. So when we focus on the lactobacillus, we choose two well-known beneficial lactobacillus species. One is Lactobacillus rhamnosus, and another is Lactobacillus reuteri. So we go further study to use a specific strains to study their interactions with these nanoparticles. We can see firstly, both of these bacteria can uptake the nanoparticles. Also, the growth of this bacteria also being infected with nanoparticles. Here from this, the lower figures, we can see the growth of bacteria being improved with the treatment of these nanoparticles. And so so far here we know the bacteria being improved and regarding their growth. So we want to know whether their products or metabolites being impacted with these particles. So we focus on the antimembrane vesicles from bacteria. From the left two figures, we can see after treatment with nanoparticles from microprotein, the bacterial OMVs being, the production of bacterial OMVs being significantly increased, as well as their particle size being impacted here, shown in the right panels. Then the production of OMV is being changed. We also focus on their bioactivity whether being changed. Here in this figure, we can just focus on the two blue bars here. The first one is the bacterial OMVs without treatment of particles. The second one is the bacterial OMVs treated with nanoparticles. Here we can see the bioactivities of nanoparticle treated OMVs being significantly improved, and that means their bioactivity is being improved here. So we go further, try to figure out the differences between these two OMVs treated with or without the microprotein-derived nanoparticles. We do some analysis regarding their metabolism. Here we can see some lipids metabolism and some lysosome, like this is cellular progress regarding about the cells endocytosis. Something is like this progress. So further, we do lipidomics to take a look at the changes between these two kinds of OMVs. Here we show that all these lipids have been significantly increased with the treatment of microprotein-derived nanoparticles. And based on our these lipidomic studies and the proteomic studies, and as well as some literature studies, so far we focus on the pathways, how the OMV, the activities of OMV is being changed. So far we mainly focus on the interaction between these OMVs with lysosome, and as well as some lysosome related pathways. We also do some validation related with lysosome activity and lysosome related pathways. I'm not going to go into very details about this part. It's still ongoing, this section. So in general, our study try to figure out the nanoparticles from microprotein impact bacteria and as well as bacteria metabolites. We focus on OMVs, and now we try to figure out why this happened and try to explain this phenomenon. So this is a very brief introduction about our study regarding the microprotein related bioactive peptides.
Wonderful. Thank you very much, Dr. Han, for joining. So sorry for the issue. If you would like to spend the next 7 minutes finishing up the rest of your talk, and then we'll make sure the recording goes out to anybody who's dropped off. And then regarding Q&A, I think we'll make sure to send it to Dr. Wu and yourself, and then also email those answers back out to the audience. We do have all the questions which came in recorded already. So let's pass back to Dr. Han. Thank you so much again for your time.
Okay, thank you. Simply. Okay, I'll continue my talk. So actually, it's maybe just the last second one slides here showed, and mainly we focus figure out the ways how this OMV is being impacted with the microprotein-derived nanoparticles and their potential anti-inflammatory pathways here. And this is just a brief introduction about our recent research regarding the microprotein-related bioactive peptides and microprotein-derived nanoparticles. So in our study, we mainly focus on the bioactive functionalities from microprotein-related components. So that's all I'll talk. Thank you, guys.
Thank you so much, Dr. Han, for that talk. I think understanding about the potential of fungal mycelia and the resulting mycoprotein and their effects on gut microbiota is actually very interesting for us. So we'll now open up the Q&A. I just want to check if the speakers, the attendees that are on the call right now have any questions for our two speakers. We did see a few questions coming in earlier. So we'd be really happy to answer the questions over the next 5 minutes. And in the meantime, I'd actually like to ask a question for Dr. Han. When using the food industry byproducts as feedstock, how do you approach standardization of input quality, and does this affect the peptide or OMV profiles?
So OM prefers, we did some analysis about their lipids compositions. So also they have maybe some bioactive components like just like polyphenols, like these things. Another important thing is the nucleic acids. This also may show some functionalities. So, but since there are too many components in the OMVs, so we so far we just focus starting with focus on the lipids composition and their changes. So as we show here, the trained OMVs their bioactivity is being improved. So, and also the lipids composition, particularly some of the like the 𝗲𝗵𝗲𝗲𝗲. So far we focus on 𝗲𝗵𝗲𝗲𝗲 kind of lipids, and it's been the levels of this kind of lipid being increased 15-fold like this. So also their bioactivity being improved. So far we focus on lipid dissections. If guys interested in this part, maybe they can try some maybe nucleic acids. It also maybe works, and lipids is also a very important part. I think so.
Okay, thank you so much, Dr. Han. We have one more question for Dr. Wu, actually. From an ingredients point of view, what are the most common blockers, and are there any pre-treatments to help with this? Are there others that won't be found using this approach? And have you done this assessment on cultivated meat before? That's a 3-part question.
Yeah,
but
there's 3 questions. Yeah. So I assume you are asking whether there's any blockers that block the digestions, I presume. So from our experience, it seemed that every ingredient has its own problem. So really, again, there's no one size fits all approach and problem. So really, when working with the ingredient, we really need to look into the ingredient and understand what is the issues. Because digestion is really a complex problem. Like you are seeing different breakdown. You are seeing solid becoming soluble. You are seeing the soluble being digested, and then the digested one becoming further cleaved to the shorter one. So we have seen then to complicate, even there are interaction with the lipids, interaction with the carbs. Sometimes proteins are fine, but they are just not possible to meet with the digestive juice. It's just like trapped inside bigger blocks of the carbs or fat. Yeah, so really depends on the ingredient. Yeah. What was the second question?
So the second one was, are there any others that won't be found using this approach?
Yeah, of course. Like for example, if it is about the physical particles, like how big or small it can break down after you're chewing, then this won't be possible to understand through the peptidomics approach. And also, like I mentioned, the nutrients interactions, like the macromolecules interactions. Yeah, this would be also something that cannot be directly accessed through the peptidomics approach. So what peptidomics approach is really suitable is for really relatively pure protein sources. So like protein ingredient, raw ingredient. When it comes to food metrics, it is not as robust compared to when studying just a raw protein form. And I have personally, I have not worked with cultivated meat, but if I would make a guess, I would guess that it will be quite digestible, since biologically it should be like same as the real muscle cells or fat cells that is already in the authentic animals food. Yeah.
Okay, thank you so much, Dr. Wu. I think any other questions that our audience have, you can reach out directly to Dr. Han or Dr. Wu or to us, and we can direct the questions to our speakers. That concludes part 2 of the All Protein Dialogues webinar on nutrition. Thank you so much to our speakers, to our attendees for your patience and bearing through our technical issues. And thank you so much for joining. I'd also like to thank my team for the support that they gave throughout the planning of this webinar. And that concludes the July edition of All Protein Dialogues. And with that, we will see you next time. Thank you so much.